Okay, thank you, moderator, for introduce me, and thank you, conference, to invite me to be a guest speaker for today. And good morning, all participants for today presentation. I would like to present in the topic of molecular aspect of emerging bacterial and viral diseases of Tilapia and barramundi culture in ASEAN countries. Before I will talk with molecular aspect, I would like all of you to know about the tilapia and the sea bass. The tilapia or Oreochromid species is the native freshwater fish in Middle East and Africa. It has expanded rapidly worldwide, and the most important food fish throughout the world is known as aquatic chicken due to commercial scale farming and its production. You can see that in all regions of the world, we consume and we have the tilapia culture, including Europe, Asia, America, Australia, and Pacific. And the second most important species for ASEAN country is ASEAN sea bass or barramundi, Latex concarifer. You can see from this graph that in our ASEAN country, we have a lot of sea bass farm more than another kind of fish like grouper farm. ASEAN sea bass is an euryhaline fish with a great commercial value in Southern Asia and the Pacific. Its culture in both earthen pond and cages in freshwater and brackish system. For fish farming, fish is the most valuable aquaculture species after shrimp. For tilapia, the red tilapia culture has become increasingly due to of its preferable market value comparative to Nile tilapia. Farmed is floating cage system. We have this kind of farm a lot in ASEAN country. This kind of farm is high stock density and poor water quality management. And also the intensive production is undoubtedly threatened by the potential devastation of infectious diseases. As you can see in the picture, the most important diseases of tilapia is the streptococcosis, columnaris, hemorrhagic septicemia, such as Aeromonas septicemia and Edwardsella septicemia, Franciscellosis, tilapia infection or tilapia leg virus infection, parasitic infections such as Trichoderma, Dactylogyrus, and mixed infection or concurrent infection that you will usually find the mixed infection a lot in fish species. This is the diagram of culture period of tilapia. You can see that in each period of cultural system, you can find a different of disease in cultural system of tilapia, including bacterial, virus, and parasitic infection. The picture shows the streptococcosis clinical signs. You can see like exophthalmos, erratic swimming, or circular swimming, because it's the streptococcus is will affect to the brain or meninges of fish. You can see exophthalmos like this. This is quite common clinical signs of streptococcosis in tilapia and also in sea bass. This is the columnaris disease. You can see the fin rot, gill necrosis, and also sudden back lesion in fish that are infected with Flavobacterium columnar. This disease is make fish high mortality. The next disease is bacterial hemorrhagic septicemia. You can see that like a lot of hemorrhage at the body and around the fin of the fish and also internal organ of the fish. Next disease is Franciscellosis caused by Francisella species. You can see the clinical lesion of Franciscellosis like white spot nodule in internal organ. Next disease is tilapia virus infection or tilapia leg virus infection. The clinical signs that you can see is a big belly of fish and exophthalmos, hemorrhage as body and around the fin area. And for histopathological lesion, you will see syncytial hepatitis in the liver. The next important species is sea bass, and this is the important diseases of sea bass. I will separate into two kind of culture system of sea bass. The first is marine culture. For marine culture sea bass, you can see streptococcosis, Tennessee baculosis, scale drop, and muscle necrosis from Vibrio species, scale drop disease virus or SDDV infection, infectious spleen and kidney necrosis virus or SKNDV infection, viral nervous necrosis, VNN virus infection, iridovirus infection, and parasitic infection. And for the freshwater culture sea bass, you can also see streptococcosis, columnaris disease, and Aromonas septicemia. The disease is fish usually infected by multiple organisms called concurrent infection, same as in tilapia. And you can see same as tilapia that in the different stage of culture system of sea bass, you can see or you can find the difference in the disease of sea bass, like in the weight of around 1 gram, you can see VNN infection. On the weight around 15 gram to 100 gram, you can see ISKNDV or iridovirus infection. Or when fish nursery or pre-grow out in the sea water or marine water, you can see Tennessee baculum maritimum infection. But if grow out in the fresh water, you can see another kind of infection like columnaris infection is caused by freshwater bacterial species. Also, when the fish in still culture in marine, you can see infection of Vibrio species and Streptococcus innae. This is the picture of clinical signs of columnaris disease in freshwater culture sea bass. You can see the lesions look the same as in tilapia, like gill necrosis, fin rot, and sudden back lesion. This is Tennessee baculosis. It's in marine culture sea bass. You can also see the clinical lesion look the same as the columnaris disease, but this one can occur only in marine water culture sea bass. This is scale drop disease virus infection or SDDV infection. You can see the clinical signs of like a lot of scale drop and muscle necrosis and fin rot and also hemorrhage around the body. The bacterial scale drop and muscle necrosis disease or SDMND that caused by Vibrio species such as Vibrio havii. Next disease is infectious spleen and kidney necrosis virus or SKNDV infection. The clinical sign that you can see is fish will get like black body, pale gill, and emaciation. This is the clinical signs of viral nervous necrosis or VNN infection. You can see the lesion at the nervous system of the fish. This is iridovirus infection. It's caused very high mortality in small fish, and when the fish get iridovirus infection, you can see like the hemorrhage in the retina of the fish, and the body of the fish may change in color. This is the streptococcosis infection. The clinical signs that you can see is the hemorrhage in the internal organ and also around the body area, around the fin, and also same as in tilapia is exophthalmos. Normally, when we would like to study the bacterial disease, we have this conceptual framework for study of it. First of all, we have to isolation and identification the isolates of bacteria from infected fish, and then after we get the bacterial isolate, we will do phenotypic classification, genotypic classification. You can see that sometimes only phenotypic classification alone cannot get the good result for the causative agent, genus, or species. Then we cannot study more or go deep in another study if we don't have a good tool or good techniques for helping in the study or in the research. So you can see that from this diagram, we need genotypic classification, or we need the molecular techniques for classification and for study another thing about the disease of fish, same as other disease in other animals. After we get the information from phenotypic classification and genotypic classification, okay, we can summarize the characteristic of bacterial species that we isolated from fish, but that is not for all. If we would like to study more and more, we need another thing. So what we need to combat the disease of the fish, we need a rapid diagnosis or rapid identification of the causative agents, especially the emerging pathogen. We need to understanding the molecular pathogenesis of the diseases. Then we can have a proper management of the diseases. We need the prevention and control by the vaccine and other strategies. We need the proper treatments, such as we have to know the drug and vaccine target for know the weakness of the pathogen that we can destroy them. As I mentioned all of this, you can see that we need the molecular aspect or molecular tools for help us to better understanding everything, to get the rapid diagnosis, to get the better understanding about the molecular pathogenesis, to get the right vaccine or drug to using for treatment and control of the disease of the agent that cause disease in fish. For the molecular aspects using for diagnosis of bacteria and viral infection, I list this for you, like polymerase chain reaction or PCR with the specific primer, the PCR with sequencing, the molecular serotyping by multiplex PCR, as we use a lot for the Streptococcus agalactiae molecular serotyping. We don't need to use the serological method for Streptococcus agalactiae, but we can use the molecular for serotyping it. We can use the quantitative real-time PCR for diagnosis of bacteria or viral infection, or we can use like another advanced techniques, molecular techniques like loop-mediated isothermal amplification or LAMP, or recombinase polymerase amplification or RPA to use for diagnosis of bacterial or viral infection for fish. Sometimes we also need like the tools or the bioinformatics tools to analyze the information that you get from the molecular aspect, like after you sequencing, you have the nucleotide sequencing of the pathogen, then you need some bioinformatic tools or basic local alignment search tool or bioinformatic tool to using for analyze the sequence that you have. I would like to show you the websites of like basic local alignment search tool. As you can see that they have many kind of basic local alignment search tool to use for analyze the sequence. This picture show that the techniques of loop mediated isothermal amplification or LAMP that I will not going deeply into the detail because we have a limited time. Just would like to show you roughly about the techniques. This is the techniques of recombinase polymerase amplification or RPA that you can use for diagnosis of bacteria or viral infection of fish. Another aspect of molecular aspect of for characterization of bacteria and viral pathogen, including the PCR plus restriction fragment length polymorphisms, PCR plus sequencing, phylogenetic analysis, multi-locus sequence typing or MLST, next generation sequencing and genome analysis, and the prediction of antigenic epitope for vaccine or target for drugs. This slide would like to show you an example of the molecular aspect that we use for characterization of bacterial pathogen. In my case, I use like 16S rRNA amplification by PCR plus with restriction fragment length polymorphisms. I use intergenic spatial region sequencing and phylogenetic analysis. I use 16S rRNA sequencing and phylogenetic analysis for using for characterize my bacterial pathogen, that my bacterial pathogen is where is distributed, or what is the genetic diversity of them, or what is the clade that they are located in. You can know everything after you use the molecular technique or molecular aspect to study of them. This one is the first technique that I have been used before, like 16S RNA amplification by PCR and restriction fragment length polymorphism. After you amplify the bacteria by 16S RNA amplification, you can cut by restriction enzyme and you run agarose gel electrophoresis. Then you can see the difference in the band pattern. After that, you can compare that is located in the same group or different group. As you can see in this picture, you can see that this Flavobacterium columnar, the fish pathogen, after run agarose gel electrophoresis, is located in the different group, group 1 and group 2 by this technique. And this is the genetic characterization using intergenic spatial regions amplification, sequencing, and do the phylogenetic analysis. You can see that the bacteria that you would like to analyze is where it's located, is which clade is located, which branch is located. Also, this one we use 16SR DNA sequencing, amplification and sequencing, and then we do the phylogenetic analysis. Then we know that the bacteria that we would like to know where is located, when compared to another bacterial pathogen or another strain where it's located. Then we can see the position that they're located. This we call genetic characterization. It's also depend on, we call discrimination power. It depend on technique, which technique you would like to use, and after testing, the discrimination power is high or low, you can select it, and I suggest to use only the high discrimination power. Then the information that you can get from the characterization will be the right one for characterize the pathogen. Then let's look at the molecular aspect using for study the bacterial and viral pathogenesis and immune responses. The techniques that you can use for a study of this one, pathogenesis and immune responses, will be including cloning techniques, sequencing, quantitative real-time PCR, molecular hybridization, including northern blot, southern blot, western blot, or even in situ hybridization, protein expression, mutational analysis, next generation sequencing. That is, they have many kind of next generation sequencing that you can select for study of your pathogen or select for study of the immune response. And after you have the information from next generation sequencing, you can use it for the genome analysis or even genome comparisons to know, to compare your bacterial or viral pathogen. Then you can have more information, deep information, that some of them you can use for like further investigation. Like you can predict the epitope of the antigenic epitope that can be used for the target of vaccines or drug, something like that. Okay, that is all for the molecular aspect that we can use for study fish disease. Anyway, I cannot go deep into the detail of each technique because the time is very limited. Then I just want to show you my research article that we published by using the molecular aspect to study in the international journal since 2012. You can see that we have the paper, like we use the PCR techniques for diagnosis, like duplex PCR for differential differential diagnosis, or for simultaneous detection of Streptococcus innae and Streptococcus agalactiae. We use the molecular aspect to do the molecular characterization and virulence gene profiling of pathogenic Streptococcus agalactiae population from tilapia. We use the molecular aspect to study what is the change or what is the regulation of the gene, even up-regulate or down-regulate of inflammatory related gene after infected with the Streptococcus agalactiae. We use the molecular aspect to study the genome sequence of Streptococcus agalactiae strain from tilapia. We can, after that, as I mentioned before, we can obtain the information to study the genome comparison, to compare the genome between pathogenic Streptococcus agalactiae isolate from tilapia in Thailand, what difference between two strains or the selected strain that you want to compare it. Yes, this is also the paper of comparative genome that any of the genome of the bacteria. You can select it for compare together. In this paper, we compare the important strain from Thailand and from Vietnam, and we see what different in it genome. Our recently publication, we use molecular technique like PCR by using the species-specific primer to specific amplification by PCR of the Flavobacterium columnar targeting chondroitin acylase gene. We use the molecular aspect by using novel recombinase polymerase amplification or RPA that I mentioned before, combined with the lateral flow dipstick assay to rapid visualization in the specific detection of Flavobacterium columnar, the causative agent of freshwater columnaris in fish. Okay, this is redundant, sorry. Okay, for all things that I present before, I can summarize that the molecular and genome study of those bacteria and viral pathogens can provide a fruitful information to the better understanding about their virulence, pathogenesis, diagnosis, epidemiology, immune response, and etc. And the information are very useful to apply in prevention and control or management of the emerging diseases, such as use in vaccine design or development, or using for drug target design and drug development. Okay, that's all for my presentation, and this is my research team. This is the institute that involved in all information or all research that I mentioned before. Thank you for your attention.